Good morning, good afternoon, good evening, and welcome to what I believe will be a truly transformative discussion. I'm genuinely excited to see the caliber of expertise we've assembled today. We have industry innovators who are redefining vaccine science, regulatory leaders navigating complex approval landscapes, and clinicians who witness the real-world burden of respiratory disease in their daily practice. We are particularly honored to have with us today an exceptional panel. Professor Osterhaus, whose groundbreaking research has shaped our understanding of respiratory viruses. Dr. Kishko, bringing invaluable industry scientific perspective to vaccine development. And Dr. Gravenstein, whose clinical insights illuminate the real-world impact we are working to address. This convergence of perspectives from fundamental research through clinical application isn't just valuable, it is essential as we examine the rapidly evolving intervention landscape for human metapneumovirus, HMPV. Let's address the fundamental question. Why HMPV and why now? HMPV isn't new to us. For over 2 decades, we've recognized it as a significant respiratory pathogen, particularly devastating for infants, older adults, and immunocompromised patients. Yet, for much of that time, it has remained overshadowed by RSV and influenza in terms of research investment and public health priority. That dynamic is changing dramatically and rapidly. We're witnessing genuine scientific momentum across multiple development platforms, followed by great recognition of HMPV as a substantial clinical burden. The conversation has fundamentally shifted from should we invest in HMPV to how do we accelerate effective development? That transformation is precisely what brings us together today. This slide captures the current clinical stage pipeline of scientific approaches. Let me guide you through them systematically. First, virus-like particle vaccines, including IVX-A12 vaccine, IVX-A12 program, now strengthened on the AstraZeneca's portfolio following the acquisition of Icosavax. Second, we have the protein subunit vaccines featuring candidates from our former ViceBio platform, which is now integrated within Sanofi, alongside Clover Biopharma's innovations. These span RSV plus HMPV combinations and extend to sophisticated trivalent RSV plus HMPV plus PIV formulations. Third, we have mRNA-based approaches, leveraging the proven flexibility of mRNA platforms through programs at both Moderna and Sanofi, creating multivalent constructs that simultaneously target RSV, HMPV, and PIV. Fourth, we have live attenuated vaccines, particularly NIAID's PIV plus HMPV combination, showing exceptional promise for pediatric populations. Fifth, we have broad-spectrum interventions, including Pulmotec's POL 042 and Synergen's SNG 001, designed to enhance host defenses against multiple respiratory pathogens rather than targeting individual viruses. And finally, we even have revolutionary cell-based therapies such as NextCell's ProTrans stem cell approach, representing an entirely novel intervention paradigm. This breadth is extraordinary, and it signals something profound. HMPV has evolved from a niche target to a central component of how we conceptualize disease prevention. What does this landscape mean for each of us? Well, for industry representatives, the implications are clear. While competition is intensifying, so is the opportunity. The most successful programs will position HMPV within integrated respiratory portfolios, not as an add-on, but as a foundational element of multivalent strategy. For regulatory partners, this platform diversity, spanning live attenuated vaccines, mRNA, and cell-based therapy, underscores the critical need for adaptive, platform-based evaluation frameworks. Scientific innovation is accelerating. Our regulatory approaches must evolve accordingly. For our clinical colleagues, this pipeline represents genuine hope, future prevention, and treatment tools that authentically reflect the disease burden you witness across diverse patient population. But here's our unifying insight. HMPV development is no longer occurring in isolation. It is being integrated into comprehensive pan-respiratory strategies, perfectly aligned with how patients experience disease and how health systems must approach prevention. Now, I'm delighted, right, to take us into the rest of the presentation. We're going to have the speakers. We have Professor Osterhaus, who will share highlights from his pioneering research that has fundamentally shaped our understanding of respiratory viral pathogenesis. Dr. Kishko will illuminate the industry perspective on translating scientific advances into viable vaccine candidates. And Dr. Gravenstein will ground our discussion in clinical reality, helping us understand how these developments will impact patient care. Together, they will explore the scientific breakthroughs, clinical considerations, and development challenges that will define the next decade of HMPV intervention strategies. I encourage you to engage actively. We're going to have time for questioning at the end of each presentation. It's just going to be about 5 minutes, but then we're going to take the rest of the questions after all the presenters are done. Thank you for joining us and for contributing your invaluable expertise to this critical conversation. So, brief introduction of Professor Osterhaus. He is the founding director of the Center of Infection Medicine and Zoonosis Research. He is also guest professor at the University of Veterinary Medicine, Hanover, Leibniz Institute of Virology, Hamburg. He co-discovered HMPV, SARS-CoV-2, among several novel viruses. His work spans over 1,000 publications with over 100 citations. He chairs the European Working Group on Influenza and the Global One Health platform, and is a member of the Dutch and German Academies of Sciences and commander of the Order of the Dutch Lion. I can go on and on about all Professor Osterhaus's accolades, but I'm going to give the floor to him to take us through.
Well, thank you very much for your very kind introduction. What I would like to do is to share my screen, and let me see if I do it like this. Can you see my screen now?
Yes.
Yes.
Okay, thank you. So what I'm going to do is, first of all, of course, I'm going to thank ISV to spend so much attention to this, what used to be new virus, and as we'll hear today, is not new at all. It has been with us for a long time. And I'm very pleased that as Chime said already, that indeed a lot of work is ongoing into this direction, as we'll hear later on. So the title of my presentation is HMPV and RSV epidemiology, so similar but still different. And there is just a little question mark there. Can I have the next slide? It doesn't seem to transport. Okay, well, these are my affiliations and potential conflict of interest. I don't think I have any conflict of interest here. So this is where the whole story started. It was just after I had joined the Erasmus Medical Center with a small virology group, and we were doing the diagnostics for the pediatricians as well. And you see here Ronald de Groot on the list here on this first paper that we published on this, and the pediatrician. And indeed, they came to us and they said, well, this diagnostics of yours, it's worthless. You know, we see so many cases of RSV and you barely ever diagnose one. And as a matter of fact, we selected 28 children that were diagnosed clinically as RSV. And we sat together, Bernadette van der Hoge, Ron Fouché, and all the other people in the lab. And then we said, so let's see what's going on. Do we have a funny strain of the virus, or what's going on? Why can't we isolate the virus? That's how the whole story began. Well, basically, we identified a new virus, or what we thought was a new virus. And what we did, is we saw that this particular virus, when we had the sequence, then we realized that this was not a human virus, it was an avian virus. Well, an avian virus in 28 children at that time. So what we did is we asked the mothers and the family of the patients, have they been in the zoo or do you have birds at home? None of that. And then, as a matter of fact, the next step we looked into the serum bank of the Erasmus Medical Center at that time, into children and older adults, older people and adults who basically had no infectious diseases or broken legs or what have you. And as you can see on the slides here, then you see after about 5 years of age, every child that had come to the hospital, every person that had come to the hospital had antibodies against this virus, quite similar to what we would see in flu and RSV. So we thought this must be a human virus. It's not an avian virus. Well, that's how the whole story started. And then doing the serology, we went to Amsterdam, different places in the Netherlands, abroad, etc., and found basically the same pattern. Well, this is kind of a historical overview of what happened. So first of all, we identified the virus in children with respiratory tract infection. Then gradually, and I'll tell you that story, we got a global recognition that this was a significant pathogen for children. We developed the diagnostics and a lot of people joined in. And we start then the epidemiological work proper there with many other groups in the world. And now, definitely since the pandemic, it has become a real hot topic, as you'll hear later. Well, first of all, what is the virus we are talking about? Well, it's a pneumovirus, as we call it today. Originally, it was classified as a paramyxovirus, but now that we have two human pneumoviruses, basically RSV and human metapneumovirus. Yeah, the only metapneumovirus we had seen before were avian pneumoviruses. So basically two pneumovirus. One we call orthopneumovirus, and the other one we call metapneumovirus. The difference between the two viruses genetically is that the gene order is different, as you see on this slide, but also metapneumovirus lacks two non-structural proteins, as you see here. Well, that has quite some implications that we won't go into today. Well, then, of course, then you see, is this really this metapneumovirus? Is it important at all? And you saw 2001 in Rotterdam identified, and it very slowly picked up here and there. And this one goes to 24. I haven't made the slide for 25 yet, but you'll see gradually it's moving up. Yeah, as it is being recognized as a real human pathogens, as you heard before from the introduction by Chime. So basically, what is happening and why is RSV exploding as it does? The reference is there in the PubMed. Well, basically, of course, as you have heard, we have a lot of intervention strategies ready to date, and is the same going to happen for HMPV? And we think, as we'll hear later today, that this is definitely the case. Well, when we compare the clinical disease with that of RSV infection, you see here, we know that all. When you talk to physicians and you talk about RSV, well, when you did that 10 years ago, they would say that's the virus of children. More and more clinicians start to realize that it is a major pathogen for older adults and immunocompromised as well. And you see here estimated rates in this case, the numbers of hospitalization and deaths among children and older adults. So those are the major groups plus the people who are immunocompromised. And you see here from a paper an inventory of what we have. When I learned about infectious diseases of the lung, it was all bacteria. And now we have the big five or even more, which are the viruses. And you see HMPV here being 6%. And I think this is a huge underestimate. Yeah, due to, and you'll see that here. Yeah, it's not diagnosed, or it's picking up at the moment. But if you would go 10 years ago, there was virtually no diagnostic for HMPV, and this was due to the limited awareness, the restricted access to diagnostic tools, and also the very many co-infections, both viral and bacterial co-infections. So once you find a bug in a person, you say that's the cause. And we know today quite well that this might be quite different. Well, what can we learn from RSV when we look at HMPV? I think, as you have seen, the genetic buildup of the virus is quite similar. Basically also it seems to be that the age groups overlap to a large extent. And if you now look at burden of disease for RSV, you see the major thing is in infants, and that's also for HMPV. When we discovered the virus, obviously that was in young children, or in infants. And then you see when indeed children get older, and then during adulthood all the way to older adults, then basically the virus is still circulating, disease is still there to a limited extent, and then in older adults it comes back again. This is true for RSV, and as you will hear later, it is largely true for HMPV as well. So when we look at immunity, you see here, and this is all adapted from the paper by Terstappen about RSV, then you see here first people have maternal antibodies, and once they lose it, then of course become more susceptible to the infection. And so basically then we have the proper immunity there, and then in older adults we see immunosenescence picking up. And the drivers of transmission, we know quite well these days that young children are a major one. And I would be surprised, although the studies is not there, that the similar thing is true for HMPV. And let's say immunization strategies, basically, they are not there for RSV, for HMPV yet. For RSV, of course, we have antibodies, we have vaccines, for maternal vaccination and for older adults, and we might well expect that the same is going to happen for HMPV. Well, just a slide where we look at percentages here of symptoms by age for community positive cases here at the day of diagnosis and 4 days later. And you see here in this study here in the JID, you see if you look at all the different scores, you see that basically they over age they get worse and worse, as we have seen also for RSV and for many of those respiratory viruses. So then let's look into the origin. I said, well, we thought initially that we were dealing with an avian virus, which apparently was not the case. And when we did some genetic analysis here, and we look at what is around today, and I'll show you some data about that later, then we see indeed that in some 250 years ago, possibly, here we see that the virus shares a common ancestor with a virus from birds, and that's basically here this particular virus here that we have here, that is the HMPV-C, which is still circulating. And now to date, so there must have been a spillover at some time from birds to humans, and then that drove the development of HMPV. We see at the bottom the Ortopneumovirus, the RSVs, and here we see the avian viruses. Interestingly, you see all the different clades and subclades, reminiscent of what we have seen also for RSV. Well, coming back to the timelines, when we look back into our serum samples, in the bank, then we saw that easily from 1985 onwards, there were antibodies there in people, in virtually all the people who we tested. And as a matter of fact, as you've seen from the previous slide, this virus must have been circulating for much longer. Well, we have the identification of the virus. Then we know in the meantime that we have two serotypes, A and B, and they can be divided into different genetic lineages. A1, the A1 lineage we know today is not circulating anymore. So we have A2, B1, B2. And from here we see there's a duplication in the G gene, something that we also see in the recent strains of RSV. What the exact meaning of that is, we're not 100% sure, and we see that actually in the strains that we isolate today. We see that happening all the time. Well, this is the slide about SARS, but it's actually to show you some of the history there. I remember we were involved in the discovery of the SARS coronavirus, and it was quite interesting. These are our colleagues from Hong Kong, which was the epicenter at that time, and they very proudly showed the virus that they had isolated from their SARS patient, from more of the SARS patients. When we got the virus, we looked into it, and we saw immediately on the basis of the sequence that this was not that this was HMPV, and we could not believe that HMPV was at the basis of such a big outbreak, because the virus is all over the place. And when we looked more carefully, interestingly, there was also a coronavirus was found, and all the medical people on the WHO team that we were participating in, they said, okay, these coronaviruses are wimpy viruses, so it's most probably is the HMPV, and it cost us some time to convince the people that the HMPV was not the cause of SARS at that time. But that's just an anecdote to show you, how the people didn't even realize there was a virus like HMPV, although it had been published 2 years ago. But the importance of the virus was not recognized. Well, coming back here to the phylogenetic tree, where do we stand today? We see the HMPV with the different clades and subclades here. You see the ABCD from the birds, and then RSV here. And you see this virus ramifies. There's many ramifications of the virus. There's a lot of epidemiological work ongoing to identify the virus, see what's going on, which is important, but it's a relatively stable virus. HMPV, like RSV, although we have different subclades, there's a lot of cross protection there. So this is a paper by the group of Bernadette van der Hogen. She was the first author on our first paper, and she spent a lot of her career on working on human metapneumovirus, and especially the epidemiology as well. And this is a lot of facts, and I'm just pick out, and this is from 2023. We have the two A and B lineages I mentioned before, and then we have the sublineages A12, B12. Then if we go to the fourth bullet here, there's no difference have been observed in the dominant lineages between the samples obtained from patients with respiratory infections from hospitalized versus general practitioner. And in both populations, we have this duplication of the duplication of the nucleotides in the G protein. Interestingly, one of the sublineages, A1, is no longer detected, as I said before, in the Netherlands, but this becomes more and more clear that this virus seems to be extinct. Well, then let's go back. What do we know through the years in terms of severity of disease? And you see here one of the early papers of comparing RSV, HMPV, and influenza virus in older adults. It was just included at that time, because they realized there was another virus and just pick it up. And so in this study, they looked specifically, and it's from Nashville, Tennessee here, where they looked into hospitalized patients, and then they looked at RSV, HMPV, and flu virus. And what they found, and you see that here by the arrows, if you look at the area under the curve over the different months, there's different amplitudes. But if you look at the area under the curve of the HMPV, you come to similar figures as RSV and influenza. And this is a picture from 2012, and it has systematically come back all the time. So I think the conclusion that we may make is that indeed RSV and HMPV are more than a threat to infants. And you'll hear later from Professor Gravenstein what indeed is this virus causing in older adults. Well, and then of course, this is the first paper that we published together with Pat Kane, where we had a stem cell patient transplant recipient who actually died from respiratory tract disease that we could diagnose as HMPV. That was the first case. And more of those data has become available in the meantime. And when we look at this slide here, and this is from a Canadian study here, from a recent Canadian study, where they looked at the human metapneumovirus epidemiology among middle-aged and older adults who were hospitalized for acute respiratory infections. And then you see indeed the message that comes out that frailty is definitely associated with severe disease. You see here comorbidities, cancer, chronic heart failure, COPD, diabetes mellitus, liver disease, less for liver disease and chronic kidney disease. But basically, it is a picture that emerges that, well, well, you'll hear later about that, but it is quite similar to what we know for the other infections. Well, some old and some new infections to make an estimate of hospitalized patients with chronic pain or with pneumonias in principle, and you see that the data ranges from 5 to 15% of those hospitalizations are due to HMPV infection and RSV. Yeah, so basically 5%, 5 to 8% for RSV. Paper from 2008, one from 2012, but also a more recent paper here from Sanofi here in the JID, also coming to 5% for HMPV. So I think that's about the range that you have to think about in severely hospitalized patients with pneumonia. Well, then we look at, and there's many reports about co-infections in the meantime. And you see here, this is the total number in this particular study, the same study here, where they looked at overall cases here, and the blue ones are the RSVs and the others are the HMPVs. And you see virus alone in both RSV and HMPV is the minority, and mixed either viral or bacterial co-infection, we see it a lot. And the tendency that's not clear from this slide is that especially for HMPV, there's a lot of co-infections occurring. Well, now looking into the epidemiology proper, if you see in the pre-pandemic period, we now speak from the pre and the post-COVID pandemic period. If you see that the peaks, and you see that for many countries, the peaks of RSV, the dotted line, and the solid line, they coincide to a large extent, although there is a tendency for HMPV to be a little bit later. Then, of course, we had the virtual absence of the infection during the pandemic. And then now the thing is, gives some complications, but we have the impression that we're going back to the pre-pandemic situation again. So basically, they, to a large extent, coincide. And this is a paper that just came out, where indeed there is an overview of changes in patterns and timing in the pre versus post-COVID area. So you see here the dark area, that's COVID. You see the different countries here, and the intensities, you see it here. And it's largely dependent on climatological changes as well. So there is a clear periodicity there with the most severe cases, or most of the cases being in the winter period, similar to flu and RSV, with a tendency for these to be a bit on the later side. Here, for instance, This is one from Germany here. An interesting study here in the season 23-24, and it's kind of typical, although it can be completely different in the year after and the year after that. But the red peak, being after the RSV peak. Well, and that as you have seen from the previous slides, I can show you many of those slides. But this again, also in India, this tends to be a winter epidemics, all the time. And you see that here in a long-term study in India as well. When you look at cases in the red line, is the cases of the percentages of positivity for HMPV. So basically, worldwide, we see this virus circulating, especially in the winter times. And you see here another manifestation of that. So both RSV and HMPV epidemic months overlap in the Northern Hemisphere. These are all Northern Hemisphere countries. And you see there is a tendency for the green lines a little bit later than the red lines, although you have a lot of overlap. And it's not always the case. So I think this is what we know. They both happen in about the same period. Then there's an interesting thing that there was this last year. There was this enormous scare in China. There was all over the newspapers, and we got a lot of media contacts saying that we have a new pandemic ongoing, and it's a new virus. And they said, and they had identified it as eventually as HMPV. It was a very bad one. And so here the Economic Times here at that time, HMPV may be rising in China, but it has been seen reported in the US previously, not realizing that we discovered that virus in 2001, and it has been around all the time. Well, if you look at the next slide here, you see HMPV, and you look at the periodicity in this 3-year period, and you see indeed that HMPV is one of the big five, and you may have more severe years and you may have less severe years. But so basically that's all in the game. So like for RSV, where you may have very severe years, we had in China at that time there was a very severe year, for HMPV. And the interesting thing is that that the periodicity or the seasonality of HMPV has completely disappeared, but then changed after the pandemic, probably due to the immunity in the population at large. Well, basically, I think I've shown you on the one hand that RSV and HMPV are very similar viruses belonging to the same Orthopneumovirus genus, and you have on the one hand the Metapneumovirus, and on the other hand the Osterhaus. Well, you see here the difference, as I've shown you before, the difference in the genome constellation, where you have the two additional non-structural proteins here. Yeah, and so basically they have very similar structures, and there's very limited antigenic drift, although like for RSV there is some drift ongoing. But since it is limited, both of these are quite interesting pathogens for vaccine intervention. Also given the severity and the incidence of the disease in both in young infants and in older adults, and to a large extent also in immunocompromised individuals. Well, this brings me to the conclusions of what I was going to talk about. It is a virus that was discovered, let's say, 25 years ago. And for a human virus, that's quite unique. A human virus with a major impact, to be discovered so basically so recently. That's something that is not being expected. And I think all the big five and all the major respiratory viruses that we know today, I'm not talking about new pandemics, but all the epidemic viruses. I see the epidemic virus, seasonal epidemic viruses, respiratory virus, they have been identified, although there's many others to be found in the future, I think. Well, as we have seen, it causes globally, that's important, very serious acute respiratory infections in the upper and lower respiratory tract in infants, older adults, and immunocompromised and frail individuals. I think we should keep in mind that the virus is circulating in the population at large as a minor pathogen as well. Well, the burden of disease is for HMPV infection is comparable to that of RSV and influenza in older adults. And there are many papers, I had no time to go into that, showing that the one is worse than the other. And this is true for HMPV, but also you can find papers the other way around. So basically, it is a major pathogen. And if you make a vaccine for RSV and flu, I think there is enough reason to do the same for HMPV and see how you can combine that. Well, we have epidemics in the very early spring. Obviously, that is in the moderate climate zones, like in the Northern Hemisphere. Most of the work has been done. Obviously, in the tropical areas, it will be different. Still no vaccine or antiviral, as we heard from the introduction at the beginning by Chime. And also no antiviral treatment. So there are a lot of monoclonal antibodies and vaccines in development at the moment. And given the similarities of the two viruses, and we'll hear about vaccine development based off the pre-fusion F protein for RSV, well, the structures of these viruses are very similar. So it would be a logical thing, given the burden of disease it causes, to do a similar thing as for RSV, and preferentially, of course, combine it and then have vaccines for the big five: SARS-CoV-2, influenza, HMPV, RSV, and possibly parainfluenza. So basically, I think that's where we are going for vaccination against more of these respiratory diseases. And as we have seen, there's several of antiviral and vaccine candidates in the pipeline, and we'll hear from that more. So this is more or less what I wanted to update you about. On the one hand, the burden of disease yeah, in different age categories on the one hand, but also that we desperately need a vaccine, and that it is not unlikely that that vaccine development will be ongoing, and especially discussions about how to combine these vaccines will be one of the major subjects of the future discussion, of course. Well, thank you very much for your attention.
Thank you very much, Professor Osterhaus. That was fantastic. We have time for a few questions, and we have questions in the chat. So the first question is HMPV. So it's thanks for the nice presentations. HMPV, like flu, may be an annual vaccine, while for now RSV is not. Can you comment on how that might impact development and use of the combination respiratory vaccines?
Oh, yeah, that's a hot potato, obviously. So flu vaccination, we have to do that every year because the virus changes all the time. And the recent studies for RSV have shown that immunity after vaccination with the current vaccines lasts longer than for one season. So we might not have to vaccinate. We probably don't have to vaccinate every year, although the protection goes down a little bit. And that's a thing we have to seriously look into, how to do that. So the ideal thing would be one shot for all the 5. Yeah, but given the fact that you then vaccinate too much against RSV, and my prediction would be, given the similarities between RSV and HMPV, that there would be a similar thing for HMPV. That if you do the same trials, yeah, and then with combination vaccines, you might well expect to have the same outcome. But it will largely depend on the type of vaccine, of course, and the possibilities to combine them. For me, the best way to do it, of course, would be one shot with the big 5 represented, but I'm afraid there's a lot to be studied there before we are there, and we might have to come to a vaccination schedule a little bit similar to what we have in the pediatric world for the geriatric world, so that we really make a scheme for, and it's actually rather talking about lifelong vaccination. Start with children, and there's young adult vaccines, there's adult vaccines, and there's older elderly vaccines. Vaccines for older adults. So I think that's the trend that we see today, but it's very difficult to predict these things because whatever you predict is not going to happen.
Indeed, very, very rapidly evolving landscape. We have a second question. With the introduction of RSV vaccines and monoclonal antibodies for children, do you think there will be population level decreases in RSV that will open the ecological niche for HMPV to cause more disease in neonates, where RSV has dominated?
Well, basically, there's first of all, there's no cross immunity between, as far as we know, there's very little antibody, if any, and very little T-cell cross immunity there. So I think these still are 2 completely independently circulating viruses. Yeah, the only thing I could imagine, if you have children infected with flu or with RSV, etc., they are less likely to be infected with HMPV, but that's only a very small minority. So I don't think these 2 will influence each other. Yeah. Also, you see that, like in RSV, the A and B do circulate independently. Yeah, and the similar thing seems to be true also for the clades and subclades of HMPV.
Wonderful. And we have someone in the audience that has raised his hand. I think Peter Palise.
Peter Palese.
Palese. Can we unmute so that he can talk?
Can you hear me?
Yes, we can. Yep.
Okay. Wonderful and I really appreciate it. You know that we have in the States problems from higher ups with respect to mRNA vaccines. In order to fight the big 5, do you think that mRNA as a platform for all of 5 is something we should try to do, despite what our powers that be in the United States say?
Yeah, I think the mRNA platform is definitely a promising platform. It's very new, obviously. Yeah, so and anything that's new that, well, apart from the situation in the US, but generally speaking, yeah, that's being regarded very suspiciously. But it was really a fantastic thing during the pandemic, and we have a lot of experience there. So I definitely think that we have to continue. And the other thing is that mRNA vaccines, an area that has not been explored for mRNA vaccines, and I think it should be done, is looking at conserved T cell epitope, because we are all focusing at the moment on antibodies. Yeah, so induction of antibodies, which is a very good correlate of protection for these viruses. But we know for flu, for instance, and we know for some of the other viruses in the meantime, that very good CTL responses against conserved proteins correlate with less severe disease. And that for that particular purpose, an mRNA vaccine would be much easier and would be much better than the protein-based vaccines, basically for the induction of a CD8 response. Basically, I think you need a presentation de novo. So I think this is a long answer. I realize that, Peter, but I think it's a wonderful technology. We haven't seen everything yet. Skepticism is always good. Skepticism has always been there for vaccines, but I wouldn't be skeptical at this moment, and I think we should explore it as much as possible.
Thank you very much, Alp.
Thank you, Peter.
Well, thank you. And we're going to have time for additional questions at the end of all the presentations. So if you have any questions for Professor Osterhaus, just type it in the Q&A box or wait till the end and you can raise your hands and we'll unmute you. So our next speaker, thank you very much, Professor Osterhaus. That was wonderful. Our next speaker is Dr. Kishko. He completed his PhD at the Morningside Graduate School of Biomedical Sciences at the University of Massachusetts. In 2011, he's a senior principal scientist and head of the pseudoviral production lab with Sanofi, based in Massachusetts. He performs vaccine discovery for respiratory viruses, including RSV, HMPV, PIV, SARS-CoV-2, with a focus on the older adult population. And he has made numerous contributions to vaccines research and development and is the author of numerous publications in international scientific journals. Dr. Kishko.
Thank you for the introduction. I think somebody needs to stop sharing so I can share my deck.
Sorry for that. It was me here.
All right. All right. Are you seeing a presenter mode or the?
So you can just change it.
Yes.
Okay.
Swap. Yep.
Right.
Perfect.
So thanks for the introduction, and very nice background was just presented, which will make my presentation much easier. The title of my talk is the same as that of our recently published manuscript, a computationally designed prefusion stabilized HMPV fusion protein vaccine antigen elicited a potent neutralization response. Quick disclaimer, myself and all the other authors are current or former employees of Sanofi, and all the studies were funded by Sanofi. There'll be three portions to my presentation. First, I'll start with a brief introduction to HMPV vaccine discovery and why we're seeking to stabilize its fusion protein in the pre-fusion conformation. Then I'll move on to the strategies we utilize to do so, the constructs we came up with, and their biophysical properties. And lastly, I'll show you the in vivo performance of these antigens. As you heard, HMPV is associated with a high disease burden in the older adult population, and currently there are no vaccines or even specific therapies available, but also, as was shown, there are a number of vaccine discovery efforts. Many of these focus on the fusion protein of HMPV. This is the protein that mediates the entry of the virus into host cells and is also the major target of the humoral immune response. That is, HMPV neutralizing antibodies are primarily directed at the fusion protein. There are two genetically distinct lineages of HMPV, A and B, that are further divided into sublineages and then can be divided even further. Most of these differences are coming from the other major surface protein of HMPV. It's attachment protein, or G, that has only about 30 to 37% sequence identity. However, the fusion protein, which I will be referring to as F from here on out, has at least 94% sequence identity at the amino acid level across all HMPV strains, making it a very attractive vaccine target. And as was mentioned, there is not much genetic drift over time. HMPV-F is initially made as a single chain precursor. It is then particularly cleaved into two subunits, F1 and F2, that are held together by a disulfide bond to form a protomer, and then three of these F1, F2 protomers come together to form the complete protein that is displayed as a trimer on the surface of the virion, initially in this mushroom-shaped metastable prefusion conformation. During the viral entry process, or also spontaneously, this form can transition to this Golf T-looking shape. This is the post-fusion form of the protein. It is much more stable, and this transition is irreversible. Now, while there is no HMPV vaccine yet, 3 RSV vaccines have recently been licensed, and all of those utilize the prefusion stabilized form of RSV-F as their sole antigen. And given how similar the RSV and HMPV-F proteins are structurally, they're so similar that they actually have about 30% sequence identity, and there have been monoclonal antibodies isolated that can neutralize both RSV and HMPV-F. It is thought that following a similar approach, that is stabilizing HMPV fusion protein in its prefusion conformation, would give the best route to a safe and effective HMPV vaccine. And in fact, in animal studies, it has been shown that immunization with prefusion stabilized HMPV-F does induce higher neutralizing antibody titers than immunization with the post-fusion form of the protein. So now that you know why we're interested in stabilizing HMPV-F in its prefusion conformation, let's take a look at how we went about doing so. The first thing we did is to select the base strain on which to base our constructs. We wanted to ensure that we use the most relevant sublineage of the 4 main ones, and to determine what to use, we simply downloaded every HMPV-F sequence with a known year of isolation and made this plot. And as you can see, in recent years, subtype A2 has predominated, with the B subtypes usually seen at lower prevalence. And as was mentioned, the subtype A1 is extinct. And therefore, we chose a prototypical subtype A2 strain, Candida 9793, as our base sequence. During COVID, there was very little HMPV sequences, mostly because of the success of the non-pharmaceutical interventions, but also because all sequencing efforts are focused on COVID. But as was shown in the previous presentation, HMPV has come back very strong post-COVID, but the patterns of subtype prevalence have stayed fairly similar to those just before the pandemic. To generate our pre-F stabilized candidates on this base strain, we utilized the computational design strategy, where we basically utilize Rosetta modeling to calculate how much each potential mutation changes the protein stability by comparing the free energy difference between the prefusion and the post-fusion shapes for both the wild type and the proposed mutant proteins, selecting the ones with lowest energy, and then doing multiple iterations of this cycle. The specific types of mutations we utilized were hydrophobic cavity filling and intra and interprotomer interface stabilizations, which I will go into details in the subsequent slides. But first, I want to mention that in addition to our in-house candidates, we also generated 3 benchmarks. The first of these, the A185P, is a prototypical prefusion stabilized HMPV-F from the McClellan laboratory. It's stabilized by this single proline helix breaker mutation. In this and the subsequent slides, I'm showing the mutated residues in these colored globes, each color representing one of the 3 protomers in the trimer, and then showing location of the mutations in the pre-fusion state and where they would be in the post-fusion. Now, this prototype was made on the A1 backbone, and as you heard, the A1 sublineage is now extinct. So to make sure we're comparing apples to apples, we also made a benchmark putting the same proline into our A2 backbone to generate this D185P construct. And lastly, also from the McLean laboratory, we took the post-fusion stabilized construct. Looking now at the specific types of mutations we introduced, our first strategy was hydrophobic cavity filling, that is exemplified by this K138F mutation. And here we're simply seeking to increase the energy barrier of the protein to unfolding from the pre to the post-fusion state by packing its core with large hydrophobic residues. In the interface stabilization strategies, we sought to identify residues which interact only in the pre-F form of the protein, but are not interacting in the post-fusion form, and then to increase the strength of these interactions either by introducing stronger hydrophobic or isoelectric interactions between them. In the intraprotemer stabilization strategy, we looked at residues that are in the same protemer, exemplified by this N46VT160F mutation, which I'll be referring to as NVTF for short. And then we have the intraprotemer strategy. By the way, here we're looking down at the top of the trimer so you can more easily visualize that these 2 residues, which are in different protomers, are interacting in the pre-fusion state, but would not interact in the post-fusion state unless we, after we increase the strength of their interactions. So utilizing these 3 strategies, we came up with 20 designs, of which we were able to successfully express 6. Luckily, at least 1 from each of the 3 stabilization strategies. In this graph, we're just looking at the yield of the proteins in milligrams per liter, and this NVTF construct that's stabilized by the intraprotemer strategy gave yields 2.5 times higher than the pre-F benchmark. We selected the highest yielding construct from each of the 3 strategies to take into further biophysical characterizations. First, looking to see whether these constructs were in the correct trimeric state and whether there was significant aggregation. We did this by size inclusion chromatography. Here, this peak represents the trimeric state, and for the pre-F benchmark and the NVTF construct, well over 90% of the proteins are indeed in the trimeric state with minimal aggregation. When we look at the interprotemer construct in green, we see a smaller peak, only about 77% of the proteins in trimeric state with a proportionally larger amount that is aggregated, whereas the cavity filling strategy was entirely aggregated. This construct has no trimeric peak at all and was therefore excluded from further analysis. The next biophysical property we examined was thermal stability. This is a critical feature as an increasing melting temperature implies that your design is more stable in its current state. Utilizing Nnenna differential scaling flowmetry, we put increasing temperature stress onto these constructs and looked at their melting temperature, which is represented by this largest peak. And you can see that the melting temperatures of our constructs are right shifted relative to the benchmark. In fact, this NVTF construct has a melting temperature almost 9 degrees above the benchmark, indicating that it is even more stable in its current shape than this pre-F benchmark. Now, we've shown that our proteins are stable in the current state, but what exactly is the current state? Are they in the pre-F conformation? Do they have all the critical antigenic epitopes preserved? To investigate this, we utilized bilayer interferometry on an octet. Here's a cartoon of the experimental setup and an exemplary trace showing the various phases such as baseline, loading, and association and dissociation. And to simplify, what we're doing is simply seeing whether our constructs and the three benchmarks are able to bind monoclonal antibodies that are specific to various epitopes on HMPVF. Those epitopes being the site 0 and site 5, which are only found on the prefusion state, here shown the peak of the trimer, or rather protomer. Also the strongly pre-F preferred site 3, the post-F preferred site 1, and then there's sites 2 and 4, which are the common region, that is, they are equally found on the pre and the post-fusion states of the protein. And when we do the binding study, we see a very clear difference in the binding pattern to the post versus the pre-F benchmarks. And our two constructs have a binding pattern that is identical to the pre-F benchmarks, indicating that they are indeed in the prefusion state and all the critical epitopes are correctly displayed, thus potentially making them good antigens. Now, we next sought to further investigate stability of our proteins, but in a different way than that I showed in the previous slide, here looking at their stability during prolonged storage at 4 degrees. First, we did a very quick study where we simply kept the protein at 4 degrees, freezing back aliquots at the indicated time points, then running size exclusion chromatography to see whether we maintain the trimeric state and whether there is an increase in aggregation over time. And while the trimer peaks did decrease slightly over time and the aggregates increased a bit, those were fairly slight changes. Most of the protein retained the trimeric state. And while it's hard to tell from these graphs, the NVTF construct is a bit more stable than this pre-F benchmark. Also, on this benchmark, you can just see the monomer peak starting to come up after 5.5 months of storage, whereas you do not see that with the benchmark. But to be able to better quantify this, we repeated the experiment, again freezing back aliquots, but this time doing an octet binding study similar to that in the previous slide, looking to see whether the key pre-acidic epitopes were preserved, and then as a control, utilizing a site 2 binding antibody. Recall site 2 is equally found in the pre and the post-fusion state of the protein. And what we're showing here is the percentage of binding to each of these epitopes normalized to the binding at the beginning of the experiment. And as you can see, all the epitopes are quite stable for the NVTF benchmark after 6 months of storage. On average, about 82% of the trimers are still in the pre-F state. So this is very promising data, but so far we've just indirectly been showing that our protein is stabilized in a pre-F state. We wanted to look at this directly, which we did by cryo-electron microscopy. Now, just like wild type HMPVF, our construct breathes. That is, it is open and flexible, making it very challenging to visualize directly by cryo-EM. However, by complexing the trimer with 2 fabs, we were able to obtain the structure for the individual monomers, the protomers in the trimer at a global resolution of 4.1 angstroms. And we can very clearly see that all of the monomers in the trimer are indeed in the pre-F state. Having shown that our constructs have good biophysical properties, we next sought to investigate their immunogenicity in vivo. This was done in a mouse study where the animals were immunized twice, 3 weeks apart via the intramuscular route with 0.5 microgram of adjuvanted protein, the proteins being the 2 in-house designs and the 3 benchmarks. And then we sought to quantify the titers of binding and neutralizing antibodies induced 2 weeks after second immunization at the peak of the immune response. Looking first at the induction of binding antibody titers, we see that the NVTF construct induced the highest titers with the least spread in the group, and the geometric mean was 4.1-fold higher than that of the pre-F benchmark, this difference being statistically significant. Looking next at the neutralizing antibody titers, we see a similar pattern with the NVTF construct inducing the highest titers with minimal spread, and the geometric means are about 3-fold higher than in the other groups, although due to the spread in some of these other groups, we were not able to demonstrate statistical significance. Now, to further explore the source of these enhanced neutralizing antibodies induced by the NVTF construct, we looked at site-specific immune responses by competitive ELISA. Basically, we're seeing whether there is antibodies in this mouse sera that compete with monoclonal antibodies binding to specific epitopes. And the ones we looked at were the pre-specific site 1 and then also, pre-specific site 0, and then the site 1 and site 2. And we see that for the less potent site 1, we have induction of similar levels of competing antibodies between the NVTF immunized mice and those immunized by the pre and the post-F benchmarks. However, looking at the pre-specific site 0 competing antibody titers, those induced by NVTF are higher than those induced by the benchmarks, as is the case for site 2 directed antibodies, also higher levels induced by our construct. So this should at least partially account for the higher neutralizing antibody titers we observed. Lastly, while HMPV-F is quite similar across the various sublineages and there is a good cross-neutralization, we wanted to explicitly show that mice immunized with this construct can indeed neutralize all HMPV subtypes. As there is no universally accepted neutralization standard for HMPV, we utilize as a benchmark pooled adult human sera. Human adults would have been naturally exposed to HMPV multiple times and presumably will have seen all the subtypes. And when we ran the mouse sera against prototypical viruses from each of the 4 sublineages, we didn't see any non-responders. That is, we had good neutralizing titers against all of these strains, which the geometric means similar to or higher than those in the adult human sera benchmark. So in summary, we utilized computational design strategy to stabilize the HMPV fusion protein in its pre-F state and found that this NVTF construct stabilized by the intraprotein interface stabilization strategy had very attractive properties. It yielded 14.4 milligrams per liter, was well over 90% intrameric state, and had a melt temperature almost 9 degrees above that of the prototypical pre-F benchmark. We also showed that it retained all the correct antigenic epitopes, and the pre-F sites were stable during prolonged storage at 4 degrees. When we put this construct into animals, we saw that it induced high binding and neutralizing antibody titers in the mouse model superior to the benchmarks, with these higher titers appearing to stem in part from increased levels of site 0 and 2 competing antibodies. And the mutation of this construct was able to induce antibodies in mice that could cross-neutralize all 4 HMPV sublineages, making this a promising vaccine candidate, paving the way to further optimization and development. That concludes my presentation, and I'll now take your questions. Thank you.
Thank you very much, Dr. Kishko. Very clear presentation. I don't see any questions related to your presentation in the chat right now, but I have a question for you. So with the graphics you showed about the response to HMPV, comparing it to the benchmark, the pre-F, It doesn't look like when you look at the post-F, right, the responses you have to HMPV, it doesn't look like they're significantly different than what you have to some of the other pre-F, right, confirmations. I know that with the MBTF, it was higher than the others, but the others seem to be on par with post-F. So can you speak to that a bit? Is that something that is a bit different between HMPV and RSV, right? With RSV, there's all this move for the pre-F because we know that it's more immunogenic, but is that really the case with HMPV?
Excellent question. So yes, with RSV, the pre-F form is significantly more immunogenic in terms of neutralizing antibody titers, and the best antibodies are directed to site 0. It's immunodominant. It has the most potent, is the most potent neutralizing epitope on RSV. HMPV structure is similar. It has a corresponding site 0, but that site is heavily glycosylated. So there's less exposure of it to the immune system, which makes it less immunodominant, with immune response more evenly spread across the other epitopes, including those that are shared between the pre and the post-F forms of the protein. There is definitely trends towards HMPV pre-F being giving better immune responses, being more protective than the post-fusion form, but those differences are slightly less obvious than is seen for RSV.
Wonderful. Thank you for the clarification. We have several questions in the chat, and we have some time for them. So early in the presentation, did you say that you were targeting immune responses to site 0 and site 5? Did you see immunoresponses against site 5 in the mouse study?
Right. So in the beginning of the presentation, I said that we want to ensure that the various epitopes were correctly preserved on our constructs, which they were. In competitive ELISA, so far we only looked at site 0, 1, and 2, but the work is ongoing to develop assays to allow us to interrogate other sites. Site 5 is relatively routine discovery, and people keep discovering new sites all the time. But yes, we will look at that mouse here once the assays are developed.
Okay. Then the next question, your cryo-EM shows a monomeric pre-F. Was the protein fused to a fold-on domain?
Yes, what we're looking here is a subunit that is stabilized by the fold-on domain.
And this is also related to that. Does the design have a fold-on trimerization domain? And do you think that may be a problem when RSV vaccines also have fold-on domain?
So as I mentioned, this subunit version of the HMPV-F is stabilized by the fold-on. There is some data from the literature that when you immunize with RSV as well as a fold-on, you're going to induce anti-fold-on antibodies. The effect of those on subsequent reimmunizations is not clear, but there are companies looking at ways to stabilize RSV and HMPV-F without this exogenous trimerization motif, or in other ways reduce induction of fold-on-directed antibodies.
Wonderful. Well, thank you very much for the presentation, Dr. Kishko. And if anyone has any additional questions, we're going to have time for further questions at the end. But right now, we're going to move on to our next speaker.
Thank you.
We have Dr. Gravenstein. He is a professor of geriatrics and director of geriatrics and palliative medicine at Brown University. He has a long-standing interest in infection and aging, including vaccines and antivirals, and especially in the context of frail elderly, like those living in long-term care settings. Dr. Gravenstein.
Thank you for that introduction, and welcome everybody. Really great presentations, tough to follow. So thank you for those. We're going to cover briefly an age sort of perspective of HMPV. And I'll start off with just a couple of slides to remind you of what Professor Osterhaus talked about. Then we'll go through shedding and transmission, clinical risk, diagnostic approaches, management strategies, and then the key takeaways. So what you saw on those early slides is that HMPV is a significant respiratory pathogen. It has a seasonality, and that it is primarily winter season and tends to lag a little bit with RSV. And you can compare it also with influenza, and the period of time is 12 to 16 weeks, whereas flu tends to be a little shorter. And then the other para-influenza viruses tend to also be a little different. Now to age. Here's two distinct studies. The left one is from the decade ending in 24, and the right one is the decade ending in 2016. The left one is in Beijing, and the right one is in Flanders, Belgium. And in the left one, so this is 35 hospitals contributing to a single laboratory, and they're looking at from their Beijing Respiratory Pathogen Surveillance Center, acute respiratory tract illness, and about 1.6% of specimens were positive for HMPV. And you can see the age distribution says this is mostly in children, and then it declines in young adult age, and then kicks up again sometime after age 50. And then it appears, at least in the late 80s or 90s, that it starts dropping off again. The Belgian study is slightly different in that spread, not so much on the left side, but on the right-hand side. And I just want to show that on the left side, you can see the range here is up to 60% of positives, whereas on the right side, it's a much smaller bump, just to 3%. But you can see in the Belgian study, it goes up with age, 50 and older. There's no drop-off at the tail end. That might be a difference in where people live and how they live. In China, much more care is done at home. In Western European and North American, there's a greater chance that you live in these closed settings with other older adults. That might affect the transmission. And so on the left side, this could be grandparenting, and on the right side, it could be something a little different from that. And that's speculation. I don't actually know, but fun to speculate about. So the first principle here is that it's seasonal and it lags a little bit. And there does seem to be an age association with, for older adults, that kick up and risk for getting infected happens sometime around age 50. And that overlaps with sort of the time when it starts becoming riskier for other respiratory viral diseases. The way this is transmitted is by respiratory secretions. And it's similar to that for RSV and human metapneumovirus. It incubates within the nasopharyngeal mucosa before spreading to the lower respiratory tract. And you've just heard about how it attaches with G and F glycoproteins that mediate epithelial cell entry. And you saw that the F protein now is a potential vaccine target. Once inoculated, it incubates for 2 to 5 days. And that inoculation may or may not become symptomatic. Viral shedding then lasts for 3 to 7 days, not the day you get inoculated, but starting a day, 2 days later when you can start detecting it. And then typically within 7 days, it stops. But in older and immunocompromised adults, it can last for weeks or much longer. So this is a point. In a third of adult and pediatric cases, they remain asymptomatic, even if they're high risk. The likelihood of being asymptomatic doesn't matter about which risk group you're in. So here's the second principle. Human metapneumovirus is often asymptomatic, much like SARS-CoV-2. Asymptomatic disease can still be transmissible. Age and risk for severe disease doesn't predict whether they're going to be asymptomatic. Let's talk about the viral shedding a little bit. In children, the shedding is thought to last typically 7 to 14 days for human metapneumovirus. With mild disease with RSV, up to 14 days, but with severe disease, they tend to shed longer, 21 days. With flu in children, 10 days or more. With SARS-CoV-2, 3 to 7 days. Now, a feature of flu and SARS is that the period of shedding may be shortened in people who have been previously vaccinated or had prior infections, underlying immunity. In adults, human metapneumovirus shedding is also up to 14 days, RSV typically up to 7, flu up to 7, and SARS-CoV-2 the same as for kids. For immunocompromised, shedding can last a lot longer. And this is up to 182 days. This was in a stem cell transplant where it was documented. And for RSV can also last for several months. Flu has a median duration of shedding of 19 days with immunocompromised and up to months. And SARS-CoV-2 can be prolonged. It varies by the specific SARS variant. There's even been cases where it's been shedding for over a year in a patient infected with HIV. Peak shedding. And the peak shedding is relevant in terms of how we think about constraining transmission. With human metapneumovirus, peak shedding is between day 6 and 11 after inoculation. And so there's this period when they are also likely most infectious. RSV tends to be a little earlier in the illness. Flu in the first few days after inoculation. For SARS, it starts 3 or 4 days after inoculation, and still a third of them are shedding a week out. So for transmission and infection prevention, there's some things to consider, like for COVID, RSV, and flu, producing aerosols, like through speaking, sneezing, and coughing. You produce particles that can have virus that can get inhaled and then inoculate. Close contact is relevant. So if somebody sneezes at you from across the room, your risk for getting infected isn't as high as if you're standing in front of them. And the more time you spend in that airspace with close contact, the more likely you are to get infected. It's more transmissible at lower humidity and colder temperatures. And I think the way this works is the particles that the aerosol particles, as they get thrown up into the air, in dry air, the aloft time is longer, so they float in the air longer, so there's a greater period of time where you can inhale them. The heaviest particle, unless they get dehumidified and become lighter, tend to drop to the ground. And that's why the 6 feet rule for SARS was relevant. If you were 20 feet away from somebody with SARS, your risk for getting infected was much less than if you were a few feet away, even if they weren't sneezing or coughing. The risk then increases with proximity, with prolonged shedding, with viral inoculum load. So if they have lots of virus in those droplets per droplet, your risk increases. It increases with host susceptibility to infection. So if you have immune senescence, immune compromised, difficulty with viral clearance, those are all features that make it more likely to get infected. Asymptomatic individuals typically shed for an average of 5 to 7 days. And unlike COVID, I'm not aware of any cases of rebound where they stop shedding in between and then start up again. So what do we do about infection control? The traditional notion ahead of the pandemic was that there are some respiratory pathogens that transmit primarily through aerosol. And the examples we would use for this are tuberculosis, measles, chickenpox. And for that, you'd need a respirator to limit transmission. And some were primarily through droplets, influenza, RSV. And for those, you would use a plain medical mask, a surgical mask. The current thinking has been a little bit more relaxed in how this is viewed. All the respiratory viruses can transmit via aerosol, and people emit respiratory particles in all sizes. Mostly aerosol has longer aloft time, and medical masks in all cases reduce transmission, But for certain diseases, the higher quality mask will work better, like TB and measles. And the policy that comes from that is that you want to have uniform respiratory precautions. From a public health standpoint, the advantage of that approach, it's a simplified message where you don't have to nuance what the person who's invoking it has to know in order to do it correctly, or they don't make excuses. If you have N95s available and they have active infections, N95s will work better. And you prioritize isolation and N95s when there's high viral loads and long-range transmission risk, like with TB, for example. Droplet-borne pathogens only, in the traditional view, risk the greatest transmission with short range, whereas aerosol-borne pathogens are frequently transmitted over long range, the traditional view. The current view is that this risk is greatest adjacent to the source. The risk diminishes with distance across the board, and risk increases with exposure time. So if you hang out in a bar with somebody, even if they have low level of viral load, the more time you spend there, the greater your risk of getting inoculated. The long range is mostly due to poor ventilation, not so much whether it's small or large droplet aerosol. And the policy that comes from this is to reinforce ventilation standards. You increase minimum standards in non-clinical spaces, use N95 when there's long encounters, and clinically you can filter the air with HEPA filters and radiate it with 222 nanometer ultraviolet light to sterilize the air as it passes through those ventilation systems. And the last here is the aerosol generating procedures that risk transmission increase are problematic in the traditional view, but in the current view, most of those procedures aren't seen anymore as being particularly high risk. Talking, exercise, heavy breathing, coughing, singing do increase risk, but not so much the procedures. So the policy change has been to reconsider concepts for concern of aerosol generating procedures, risk stratify based on viral load, illness severity, and in the encounter length. So if you're doing something that's a very short encounter length, the having critical N95 mask is a little less important. So the principle that emerges from this is that human metapneumovirus can be shed for days or weeks in older adults. It spreads like other viruses, and spread can be mitigated in much the same way. Human metapneumovirus shares risk factors for severe disease. That risk is increasing age. It's also in places where you have increased transmission because of opportunity, and when you live in close settings like long-term care facilities. High transmission rates due to close contact, possibly higher viral load on inoculation. And the close contact here is if you need to live in a long-term care facility, it also implies that you have higher caregiver exposure, somebody who attends to you and helps take care of your needs. And that exposure is a specific exposure risk when that individual is infected. They run a greater risk of infecting you. And they often aren't just caring for that one person, they're usually caring for others. And so they have an opportunity to introduce it not to one, but several people at once to set up a case for community spread in that long-term care community. Because the people that live in this environment have underlying diseases, and because the viral load, if they get that prolonged caregiving activity, that increases their risk for pneumonia. It also, because of their underlying diseases and immune compromise, may shed longer and thereby also prolong the transmission risk. In these settings, the comorbidities are create risk that are risk for any age, which include underlying renal disease, cardiac disease, pulmonary disease, and malignancy. Immune suppression, especially hematologic malignancy, stem cell transplant recipients, solid transplant recipients, have a greater risk for getting infected and prolonged duration of shedding. And then there's a risk for co-infection with other viruses. So what gives you a severe outcome? I'm going to highlight just two papers here. One is a systematic review in adults, including those at high risk for human metapneumovirus. It looked at 119 articles for 600,000 patients, of which about 3.5% were symptomatic annually. It's a 4.3% in high-risk group. 51% of these high risk were hospitalized, 7% of them ended up in the intensive care unit, and 9% of these died. One-sided study in the long-term care facilities indicated that human metapneumovirus accounted for 21% of respiratory illness cases, with confirmed etiology in 3 consecutive seasons from 2016 to 2019. In a study that we published, residents of long-term care facilities share risk factors in addition to age that may exacerbate risk for severe outcomes like hospitalization and death. A quarter of these were hospitalized or died in that study, similar to that for influenza and RSV. So this is that study again. It's a retrospective case-matched cohort. These were nursing homes for U.S. veterans from 2017 to 2024. It's a proportional hazard for hospitalization or death. We had 173 cases of human metapneumovirus, 437 of RSV, and 746 of flu in these 132 nursing homes. They had similar 90-day outcomes with a 24% risk of hospitalization or death. And here you can see the curves over time, with the dashed line being human metapneumovirus, RSV here in the solid line in comparison, and influenza here in the solid line in comparison. So principle number 4 here is human metapneumovirus shares risk factors for severe disease with other respiratory viruses. Age and underlying disease and impaired immunity contribute to that risk. This is a slide on influenza, and I'm showing it in part because it looks like how it looks to me for human metapneumovirus and RSV. And essentially, what you should be seeing here is that sometime after the age of 50, there's an inflection point where the risk of dying or being hospitalized, the yellow for hospitalization, red for dying, increases with age. And that's true across these various respiratory viruses, human metapneumovirus, RSV, and SARS-CoV-2. So why is that? What is it about age? This is a study that was done decades ago in 1975. And what I like about it is the elegance of the approach. This study was done in mice. These mice that were studied were all neonatal mice. So in every group, it's a neonatal mouse. And what Hirakawa did was he took out the thymus in all but the first column. The control mice had their native neonatal thymus. And then he reimplanted a thymus from another mouse, a neonatal mouse, or a 3-month-old mouse, or a 6-month-old mouse, or an 18-month-old mouse, or a 33-month-old mouse, or no thymus back at all. So they were athymic. And then he looked at antibody responses to an antigen. And then essentially what he showed was with progressively older thymuses in neonatal mice, the antibody response declined. So this is showing T-dependent antibody response declining with age. And think about this 33-month-old mouse being the equivalent of an 80 or 90-year-old person. So much of that decline in antibody responses occurs due to thymic involution. And the thymic cells also produce cytokines. Some of these are produced in other places, and they change symptomatic presentation. So for example, as we get older, you get an infection, you make less interleukin-6 in response to that infection. Interleukin-6 is a driver for fever, and it helps with viral clearance. So in older adults, you don't tend to be as febrile, and you don't clear viruses as efficiently. As you get older, the ciliated cells in the respiratory tract don't work as well. And that means you cough a little less, and you have less efficient viral clearance and less efficient mucus clearance. When you get infected, the cells that produce those cytokines don't produce it as readily. They produce it for a longer period of time, and they don't abate quite as quickly. And that means the cytokines, which can drive symptoms like fever, malaise, myalgias, don't produce as profound symptoms, even if the infection ends up killing you. So that because of the delayed cytokine normalization, they tend to stay sick longer, And even 2, 3, 4 weeks out, when a 20 or 30-year-old might be fully recovered, an 80-year-old might still feel malaise and be relatively inactive. That's a prolonged pro-inflammatory state. There's less T cell help with age, and that means they have reduced response to infection and vaccination, and it's less durable, and that creates a problem with T-dependent vaccines. The last of these is that as the brains get older, the blood-brain barrier also gets impaired, and that means when those cytokines get produced with infection, they're more likely to have confusion, and that can translate into delirium, changes in appetite disturbance that occurs with the cytokine storm, or at least the cytokine bump that you get with infection in late life. So if you look at this with presentation with flu, and I think it operates in a similar way for RSV and human metapneumovirus, in older adults, you get a little less cough, you get a lot less fever, you might get the same amount of myalgia, but it might last longer, get less headache, you get more malaise that lasts much longer, you get a little less sore throat, maybe less congestion, maybe some GI symptoms that are in older adults, but not so much in younger adults. This is a paper we published early in the pandemic. When the CDC wrote the rules for who should be tested for SARS-CoV-2, their initial guidance was anybody who has a temperature greater than 38. We geriatricians thought 38 was pretty hot for an old person, and most of our older patients never actually made it to 38. That was our what we believed to be true. Because we had access to electronic health record data, and because this was the pandemic and the way we collected data changed, we were able to do this unique once-in-a-lifetime study. In March of 2020, when the pandemic was declared in the US, in the VA nursing homes, they decided that all residents should start getting temperatures assessed at least once a day, if not several times a day. And that meant that going forward, and that was true for several years, we had a temperature on every nursing home resident every day. Now, normally in a nursing home, you might check it once a week or less often prior to the pandemic, but this was a new day. The second thing that happened was within the VA. They decided, the VA leadership decided that everybody in those nursing homes, 132 nursing homes, should be tested for SARS on or about April 15th of 2020. So we had 8,000 people living in those nursing homes, all of them in theory asymptomatic, all of them getting daily temperatures from over a month earlier, who are now going to get tested by a molecular test on roughly the same day, sometime in the course of their illness or having no illness at all. On that day, we had nearly 700 that were RT-PCR positive for SARS-CoV-2. And then we looked at their temperatures forward and backward and aligned their Tmax over the 2 weeks before and after their positive test. So this is the alignment of their maximum temperature by 38.5 up here, 38 here. And what we showed was that only a quarter of them ever in the course of their illness, which was positive anywhere along this stretch, ever had a temperature over 38. And 3/4 of them never passed 37.2. So this is this line right here. So this line here are the people who developed no temperature spike, and the thin blue line is the control population that was test negative on that date. So this meant that that temperature threshold would have missed 3/4 of the population, and that changed the CDC guidance to think about it differently. We published one other study in this space that relates to this, and that was, we noticed that as people became infected with SARS, and I think this is true again with the other respiratory infections, that they had some temperature volatility. And what I mean by that is that their temperatures became less stable. So this is the reality is right here of what we saw in that same population you just saw a second ago, and what their temperature was at the time they tested positive. And then we looked backward from that positive test as to how their temperatures had been in the week prior to that. And the shaded gray line was when they had temperature volatility, where their temperature varied by 0.7 degrees centigrade or more on a given day, versus 0.4 degrees centigrade at baseline, versus a threshold, hitting a threshold of 37.2. So instead of having a 38-degree threshold, if we had had a 37.2 degree threshold, we could have identified a third of them a day earlier, 20% 2 days earlier, 15% 3 days earlier. But if we used temperature volatility, we could find a quarter of them even 4 days before the time when they tested positive for that round. So this isn't a great way to do it clinically, because when you get called by the nurse at the nursing home and says, "I'm worried about a patient," it's hard for them to figure out if there's increased temperature volatility. But if you're working in an electronic health record system, you could do an approach where the record itself is attached to AI that can find temperature volatility and figure out who's at risk and maybe should be under closer observation or even get tested. So the principle that comes off from this is older adults often present with attenuated symptoms, and that's thanks to immune senescence, and that's in part because many of the symptoms are cytokine driven. Certainly for RSV and flu and human metapneumovirus, the virus pretty much stays in the respiratory tract, whereas with SARS, it goes systemic, driving symptoms in a slightly different way. The symptom presentation near the onset also doesn't predict how sick you're going to get, and that's especially true in older adults. So that's a lesson for when you're thinking about antivirals, like with influenza and SARS. If you wait for them to get particularly sick, the antivirals become less effective. And if you're thinking about a medication to prevent more severe outcomes, the antivirals have to be considered earlier. There's fever attenuation, and that's a feature of advanced age, and that's at least partly cytokine dependent. Let's talk about clinical course. Using a global epidemiologic study in adults that were hospitalized with acute respiratory infection, this is work done by Falsey and others, across 12 countries, 40 centers that were hospitalized. So this is hospitalized acute respiratory tract infection. All of them consented. They were assessed within 24 hours of the time being hospitalized with a clinical severity score, and then twice after using a respiratory symptom index and quality of life measures. And of these, 635 had flu, 248 had RSV, and 107 had human metapneumovirus. The severity scores were worse for RSV and human metapneumovirus than for flu, and it includes more severe lower respiratory tract signs and symptoms of dyspnea, rales, and wheezing. The symptoms that were collected on the RRIQ for cough, fatigue, and dyspnea were really close to what the clinical ratings were for severity. So this is that study again. And you can see think about this as the severe outcomes is number 3. And here's RSV and human metapneumovirus with flu and other things. And they have slightly more severe lower respiratory tract findings than flu for a period of this study. For upper respiratory, they look relatively similar. And for general symptoms, including shortness of breath and sputum, relatively similar, maybe a little bit more shortness of breath in RSV and human metapneumovirus, corresponding with the lower respiratory tract symptoms over here. Sorry about this, is a little blurry. This is the data now. What proportion of people had dyspnea and rhonchi, yes or no, if they had an underlying risk factor? So risk factors increase the likelihood of having lower respiratory tract symptoms, these 3. Not so much for the upper respiratory tract symptoms. And yes, lower respiratory tract for shortness of breath, but not so much the other ones. This is now the same thing now divided by age, where orange is the young adults and the purple is 75 and older. And what you see for lower respiratory tract, greater proportion for 65 plus and 70 plus than the younger group. not that much different for upper respiratory tract, not that much different for general symptoms. And what I take from this is that these clinical findings, you can't really make a clinical diagnosis of somebody based on do they wheeze or not wheeze, do they have a cough or not have a cough, to say, oh, this is likely human metapneumovirus. So even though HMPV and RSV might have had more wheezing, you could get wheezing with flu. To distinguish them, you really need to do a test. That means the test you want to do needs to help drive what your clinical decision making is. SARS-CoV-2 and flu have antivirals available that can affect the outcome. And so specifically, you would like to have this on your molecular test. It might drive your isolation precautions. It might drive how the duration of shedding and whether you quarantine and for how long. So having the other viruses on a respiratory panel can be helpful to decide how to tackle this. And if you're in a setting where there's a high risk for transmission for other high-risk individuals, it might help you even more, specifically like long-term care, or when you have a caregiver that's at home and whether they need to mask when they get sick if they can't be relieved of caregiving responsibilities. Antigen testing for flu and RSV is not a great test. It's 50% or less sensitive. So molecular testing is really the best way to do this. That's true, especially for SARS and RSV. So a molecular test would be the ideal test to do. There are NAT and PCR, and they can help you with prognostication and whether you want to consider contact tracing. When you're in a nursing home, for example, and you have 2 individuals or more living in the same room and one gets sick, do you want to take all the ones that aren't clinically sick and put them someplace else, or take the person who's sick and put them someplace else? But how do you know if one of the other ones is not infected if 30 or 40% might be asymptomatic? So the only way you can figure that out would be to do additional testing, and that might help you decide how to quarantine and how to mask. Masking is not an easy thing to do in long-term care settings. This is their home, and getting them to wear it over their nose as opposed to under their chin, and their right to autonomy in their home and so forth are all issues that make care there for a respiratory epidemic challenging. In any case, supportive care is available, and that includes use of antipyretics, analgesia, hydration, nutrition. You do guideline-directed care for underlying conditions. You treat the complications. You try to get them up and out of bed as quickly as you can. You work to prevent delirium, and that's deprescribing the non-essential sedating and anticholinergic medications. And if they're not up to date with their other vaccines, you get them up to date, and you do that as a policy at the facility level if it's a nursing home, and masking. And then you hope for a vaccine. So here's the summary. Human metapneumovirus is seasonal. It's clinically indistinguishable from other respiratory viruses. Shedding, transmission may be prolonged, especially in older immunocompromised adults. It's clinically indistinguishable from other respiratory viruses, and that's the case for molecular testing. If it's available to you, it's the best way to do it. Severe outcomes are not uncommon, especially among those with the greatest risk factors. And that concludes my presentation.
Thank you very much, Dr. Gravenstein. Excellent presentation. Thank you for going through the clinical considerations. I have a question regarding the symptoms, right? You pointed out that with the older adults, sometimes you don't have the kind of severity that you would see in the pediatric population. So if thinking about some of my industry colleagues on the call, as well as myself, when designing clinical trials, right, to capture the presentation in older adults, would you recommend that some thought is given to the duration of disease as opposed to just the symptoms presented?
Yeah, I think the question you're asking is different than the way clinicians typically think about it, because you're asking for an endpoint for establishing whether a vaccine is important, as opposed to the question that I was addressing was, how do you identify the disease and then tackle it? So I think the vaccine efficacy question is a really important one. And it's also an important one in the conversation we have with our audiences. I was doing a presentation a few weeks ago where somebody said something to the effect of, if the vaccine doesn't work half the time, think about this year's flu vaccine, why bother giving it? And the answer to that question was, of course you still give it, because you get benefit even if it's an attenuated response. I didn't show a slide where when you have a bad match vaccine, like can happen with flu, that you still can attenuate and eliminate fever in a fair number of them and reduce severity of disease. So in an HMPV clinical trial, I think you want to do two things. One of them is you want to identify the number of people infected, a slightly different question, but it speaks to the epidemiologic features. And the second is the cumulative burden of the disease, which isn't just hospitalization and death. That's clearly the most expensive one. But it's also functional impairment and so forth. In looking through the literature to say, well, how often when somebody got HMPV were they no longer able to fill in the blank, get out of bed, do their personal care, and other kinds of things? That wasn't captured in these studies. And I think that that's probably a more important feature. The tail end of what happens, as you saw in the Chinese study and the Flanders study, was in late life that uptick in infection probably isn't because they're more likely to get infected because of the virus or something. It's probably because they're more likely to get infected because of exposure. In late life, your grandkids come around, you have a caregiver who comes around, and when you get infected, you just present clinically, which might not happen in younger adults who typically don't get particularly sick from this.
Thank you. And I don't see any other questions in the chat specifically for Dr. Gravenstein, but I have some questions from the other presentations. So for Professor Osterhaus, it says, you're on mute. You're mute.
I still have a question for Dr. Gravenstein.
Oh, there you go. Okay, please.
Yeah, it was a very nice presentation and given a very good overview. One of the points I am always struggling with, if you look at clinical management of cases, then when you do the normal diagnostics, yeah, you don't realize that often we are dealing with co-infections, especially with HMPV. It's also the case for RSV. But sometimes we get diagnoses from patients where we look into the virus, we do deep sequencing, and we see a plethora of different things. And then the question is really, was it really HMPV that was the bad bug, yes or no? And in all these overviews, obviously, in many of the studies, the whole issue of bacterial co-infections, let alone viral co-infections, is barely addressed. What's your opinion there?
Yeah, so in these studies, most of them would have a few percent that were diagnosed with co-infections.
Yeah.
And I think there's two parts, so I'm going to do a little digression, as my president would call it, the weave. The Staph aureus pneumonia seems to pretty much exclusively follow flu. Okay, that's not co-infection, that's a subsequent infection. And now back to your question, and that is the lag with human metapneumovirus might have something to do with making the lungs more susceptible to something when they have some other exposure early on. I'm speculating, I don't actually know. Again, now back to your question about concurrent infection at the same time. I think that the concurrent infection is a greater risk with RSV and flu than it is with human metapneumovirus and the other ones, but I think it occurs with all of them. And that has to do with seasonal overlap. So I think the value of having a multiplex PCR is that it gives you better information in real time. So what I've seen in clinical practice, when somebody, last year we had, most of the nursing homes would have antigen tests available because they're cheap. And they'd go test somebody and be negative. And the person continues to get sick and they don't bother testing again because they don't have COVID. So who cares, right? And it would get missed. And if they had done a molecular test in the first place, they probably would have found it. but they missed the opportunity. A second thing that we observed was there are some infections where they'll remember that they had the infection, and others where they won't. So, for example, the recall of having a SARS infection among our nursing home staff was very high, but they barely remember that they had an RSV outbreak a month later. And I think that has to do with the way they think about it and have to deal with it. With SARS, it's been a fire alarm now for 4 years running, 5 years running. And with the other infections, they're just figuring out that these are actually real pathogens that cause real disease, and it can end up in hospitalizations, heart attacks, strokes, maybe even dementia, as you've probably learned. So I think the opportunity is of two varieties, to recognize that they can occur and that a positive test isn't quite a get out of jail free result. It's an incomplete result. And the second thing is that not until we start testing will we really understand what the burden of disease is. And, think about, how many cases of HMPV we saw before 2001, right? If you don't test for it, you don't find it. And since then, as the apparent incidence has been increasing, I'm guessing not so much increasing, it's just we're finding it more often as we are better at testing for it.
Yeah. I agree. And the point I was trying to make is indeed, if people come with a positive RSV or HMPV test, and haven't looked at the possible other things, so then you have to be very careful to blame this particular virus on the complete symptomatology. But I think in principle we agree there. So I think, what is the future of diagnostic? That's the real question. Yeah, should we deep sequence all individuals, all samples, and it becomes cheaper and cheaper? We might well go that direction.
Yeah, I think in clinical, in closed settings where you have risk of transmission and consequence of risk transmission where it's high, I think doing multiplex testing is the right way to do it. If we can afford to do it, that's how we should do it.
Yeah.
In hospitals, probably that's the way you want to do it, because you're putting them in a high-risk environment where transmission becomes increasingly important.
Yeah.
If you have people who attend high-risk transmission settings like pediatric or adult daycare, I think multiplex testing is the right way to do it. If they're living alone at home, maybe not so important.
Agree.
Great. So the question is for Professor Osterhaus. It says, early in your presentation, you said there was a significant cross-protection between RSV and HMPV, and then later said that there's very little cross-neutralization. Can you clarify these statements? Says, yeah.
I hope I didn't say the first thing. I said basically, we can say there is no cross-protection between the two viruses, although some conserved T-cell epitopes have been found, but that doesn't say anything about cross-protection. So in principle, we can say there's no cross-protection between HMPV and RSV in principle. At least no immunological cross-protection. So that may have been a misunderstanding. Yeah, so I might not have been clear enough, but what I wanted to say indeed is that if you do neutralization assays, there is no cross-neutralization, and also at the T-cell level, there's probably very little cross-protection. Is that clear enough?
Well, it sounds clear. And if I think it was Luis Masario, if you're still around and you want to come off mute, you can raise your hand and we can be off mute. But I'll go on to the next question. So we have for Dr. Kishko, unlike RSV, pre-F stabilization of HMPV-F provides only modest immunological gains. Do you think it could still provide an advantage in the context of a multivalent or pneumovirus vaccine design?
So yes, I'll back up slightly. I think what the question is alluding to is that the difference between the pre and the post-F forms for HMPV immunization is less striking than is the case with RSV. So we're talking about the additive effect of immunization with pre-fusion stabilized HMPV-F over that with a post-fusion, which, as I talked earlier about the glycosylation, there was already a fairly good response to the post-fusion state as well. So we're, by stabilizing the pre-fusion form, we're going, adding to the baseline of the post-F. So that should indeed provide a good vaccine that could potentially be combined with those for other respiratory viral pathogens.
And then another question for you, Dr. Kishko. I was wondering if you would also expect the MVTF-induced cross-subtype neutralizing antibodies against recently circulating HMPV subtypes, in addition to the prototypical HMPV subtypes, excluding A1.
Excellent question. We did extensive in silico analysis, and there is really no significant change in the critical F epitopes. There is very little genetic drift, and most of the differences are found in proteins like G, where there was that recent deletion reported, but that protein would not be part of the vaccine.
And the final question, if there is no cross-neutralization between RSV and HMPV, is there any enhancement? Did anybody look at that?
Well, I think the question is for me, and of course we have been quite aware of problems with enhancement in coronaviruses, but in flavivirus, etc. As far as I know, this has never been shown in principle. Yeah, so that pre-exposure to HMPV would make you more susceptible to RSV or vice versa. I don't think so. So the answer is no. Yeah.
Wonderful. Well, thank you everyone, and thank you to all our panelists for this excellent symposium, and hopefully we'll have a follow-up session later on in the year where we can potentially talk about clinical data. Thank you.
Thank you.
Thank you.
Bye.